I'm Jane Srivastava. I am the Flow Cytometry Core Director at the Gladstone Institutes, which is affiliated with UCSF, or you may have had a talk from somebody from us before. I'd just like to share my screen with you because I have a presentation. Hopefully this works. Okay, so today I'm going to be presenting about a little bit about my career, but really about what a technician is and what they do. I consider myself a career technician, which is unusual in science to hear someone say that, but it's something that I'm very passionate about, is being in the technical side of science. Also, I work in something called flow cytometry, and if I ever say to anybody that I work in flow cytometry, nobody knows what it is. Even some people that work in science don't know what it is. So I'll do a bit of explanation about that. Thank you so much for the questions that you sent. I thought they were really great. I've tried to incorporate a lot of them in this talk today, and also some of them I've put at the end. But if there is anything else after the talk, please feel free to email me. I really don't mind answering them. So technician really, technical people are all around us. So a technician is anybody that does something with their hands. As you can see in the description, it's a person skilled or an expert in an art or a craft. And usually when you think of technicians, you think of science technicians or somebody that works in computing. As you can see, an emergency medical technician down there on the left, or somebody that works in a pharmacy. But technicians are also people like musicians or plumbers and electricians, hairdressers, carpenters, anybody, as I said before, that does something with their hands will be considered a technician. So in terms of science and in the USA today, there's a great structure to a technical pathway. So you have research technologists that work in cores, or research associates that work with groups, people like me, or people that go into lab management or operations management, and a lot of technicians are in the education side of science. But if you look at the history of technicians, it isn't so great. They aren't so well acknowledged. So there was a great article written by this guy, Stephen Shapin, about how technicians were ignored by the people that manage them, who were absent from published literature. Even up until recently, technicians were not allowed to publish anything. Were definitely passed over by historians and given these kind of less than complimentary names like workmen or servants or things like that. And if you think of, so Galileo Galilei, I think everybody maybe has heard of Galileo. He's got, huge pages, screeds of pages on Google about him, and is given names such as father of observational astronomy, etc. But not many people will have heard of his assistant or his technician, Vincenzo Viviani. Sorry if I butchered that one. He was also a real gifted mathematician, and he was a big proponent of Galileo's works, and he's actually buried alongside him in Florence, but nobody's ever heard of him, right? But are academics and technicians completely different? Are they connected? Can one live without each other? Or is one favoured over each other? I think the answers to that is a lot of yes and no's. There's a lot of different aspects or different things that sort of come in the mix about technicians. So I'm going to leave it. My talk's quite long, so I'm going to leave the questions in the chat to the end, but I will get back to them, I promise. But a big lab always has a mix of academics and technicians. And you'll find that many technicians actually have academic degrees, and many technicians study academic degrees when in a technical role. Some people will chop and change between technical and academic roles over their career, but I consider it a lot like this sort of multicolored rope on the side, is that everybody has their own ways of doing things and their own expertise, but altogether it makes a stronger rope or a stronger lab when the technicians and the academics are both supported really well in their career. So my own technical career, and just because you're a lab manager doesn't mean you don't draw the short straw and get put in a cabinet with a piece of equipment. I was an inch shorter than my colleague, so I got put in the cabinet to tie it up for a decontamination. So I'm from New Zealand. I went to the University of Canterbury there. I was the first person in my family to actually graduate school and the first person to go to university, which was quite fun. I really enjoyed the atmosphere of it. And I majored in microbiology. However, I have never worked in microbiology. And I think that's a big takeaway, is that, yes, you may do something at university and then you may go ahead and be in it for the rest of your life. But I think not to feel bad if you do something at university and realize then that you want to do something else, or go in a different direction. So yes, I've never worked in it, that's for sure. I have used it to get positions, and I've used some of the science I learned there, but I've never worked in it myself. My first job as a technician, but I didn't, even in going into this job, I never really thought about being a technician, as it were. I just needed a job to pay my bills. I became a research assistant in molecular pathology in my hometown of Christchurch in New Zealand. And what I did was recruited patients from an arthritis clinic and tested their blood for mutations. You can see on the bottom this gel with the black and white lines, and you'll hear a lot in at the moment in the news about the COVID testing being PCR tests. So that's very similar to what we did, or what I did, as it were. I loved this job. I loved speaking to people. I loved getting their blood and using all the equipment and getting the results out. I thought it was really great. But as I say, even then, I wasn't really sure if I wanted to stay in this kind of area, or if I wanted to head into academia or something else. So then I made a move into flow cytometry, and there was a really great question where somebody said, what made you interested in getting into flow cytometry, and what do you like about it, and things like that. I had never heard of it. I had no idea. But the other half of the job was actually what I'd done a lot of. And so they hired me for that. And then I learned about flow cytometry in the field, as it were. So another piece of advice from me is that even if there's a job that you're looking at has something that you've never done before, still apply for it. It's a great learning curve. They may hire you anyway, and then you can learn about it, which is something very, very cool. And I got to play with this, so you can see this big machine here. It looks like something from the Starship Enterprise, which is quite cool. This is obsolete now. You won't find these anywhere, except for they did sink a few out in the ocean to create natural coral reefs or something like that. I transitioned into, sorry, was someone going to ask a question? Nobody there? No, it's cool. So I did transition into lab management at the University College of London after a few years working in various flow labs. I think they hired me for my flow lab experience. And then once again, I learned about being an assistant lab manager there. So I did a lot of courses and things like radioactivity and lasers and pressure vessels and fire management and safety and stuff. So that was a really great job. And then I finally went into core management, so actually managing a flow cytometry facility at Imperial College in London. I spent 12 years of my life in that building, actually, and it was a great 12 years, but no longer there. So when I started, there was only one working machine, so only one working cytometer. There was me, and there was about, I think there was about 10 people using it all. So by the time I left, there were 8 machines and a few of us and about 200 people coming to use our facility. I'm not going to go through all that stuff on the right side about teaching and management, but that's a lot of what I did. And then now I'm at the Gladstone Institutes, which is in Mission Bay. And a lot of my day-to-day work, somebody asked about that, is of operation and maintaining all the equipment and education. So I do a lot of training of people and making sure that they can use the equipment properly. And that's not only for them, it's also for us to make sure that they don't break stuff, because it's worth quite a lot of money. But also in the mix is I do a lot of finance work, which is definitely not my forte. I'm always asking people for help on that. So yeah, don't feel ashamed for asking for people for help. I do a lot of promoting of our core, because we want companies to come and use us as well as the scientists at Gladstone and UCSF. We do a lot with safety, because we actually do a lot of research with HIV and Hep C and now the coronavirus. So a lot of my work is making sure that the safety mechanisms take into account that. I grant write for new equipment, and I do a little bit of research myself. I'm starting to. So one of the big things that I am very passionate about in my job is teaching. At Imperial College, I did a postgraduate diploma in university teaching. And when I was there, somebody sent me this, and I thought it was really perfect because this completely sums up my university teaching back in the 90s. This was. So you can see the guy up the front and he's lecturing, and then you get the people at the front that are paying attention. And then in the back, you've got people chatting amongst themselves. And I think one guy right at the back is checking out that lady there. And then that's me. See that guy in the blue, and he's sitting there with his head on his hands because he just has no idea. That was pretty much me at university. It was a little bit of a rough ride, although the atmosphere was fun. And so I bear this in mind when I think about my teaching, is that somebody standing there talking at you, like I'm doing now really, is not hugely the best way to learn. So I'm developing a lot of my teaching practices to make them much more proactive and interactive with the students that I teach. So flow cytometry, what on earth is it? I like to call it Dark Microscopy's Trial just purely because that sounds quite like a cool sci-fi title. But let's have a look at what it actually is. So it developed in about the first rudimentary flow cytometer was in about the 1930s, and it was developed through microscopy. So when you look down a microscope, you're generally looking down a bright field microscope where you have your direct observation when the transmitted white light that's coming from the light source. But dark field microscopy is indirect observation through scattering of light. So this is a starfish embryo, I believe. And so you can see the pictures are quite dramatically different, but I think the dark field one gives you more sort of depth and complexity, and that's a little bit about what flow does. So when you look at the word cytometry, which if you pop it into a Word document, always spell checks it, you can actually divide it up into two. So cyto is the Greek word for cell, and then metry is measurement, obviously. So it's a cell measurement. And if you look at this process, I mean, this is a bit too much probably to look at, but it's a process where you're generating single cells, where you're getting cells from blood or from tissue or wherever, and you're putting them into a single cell suspension, and you're measuring them for fluorescence or light scattering properties. The flow bit is that they're flowing in a stream of salty liquid, and then the cytometry is that we're measuring them. So it's quite a good description. But if you really want a good way to think about how flow cytometry and microscopy are related, you can see that they've got a lot of similarities there. So on the right-hand side is a flow cytometer, and on the left, obviously, is a microscope. So they both have things like lens and fluorescence and filters in common. But if you think about a microscope and think about the cells that you're looking at, you're looking down on a car park and looking at the cars there. So you can see how many there are. You can see how big they are, what color they are, how close they are. If one person from a car is reaching out and grabbing hold of somebody from another car, if there's kids rolling around in the back, like mine usually are. But when you're looking at a flow cytometer, if you picture yourself on a bridge standing over a freeway, and there's been some kind of mild accident where nobody was hurt, obviously, but all the cars are being funneled just into one lane, and they're going past you at a big speed, but you can still see what color they are, you can still see how big they are, you can still see the kids inside, but you won't get the information about who's holding hands with each other or how close they are, but you'll get a lot more information in terms of amount. So you can look at cells under a microscope. I did have another colleague from New Zealand that looked at 900,000 cells in 5 days. He actually brought his guitar into the lab and strummed as he counted the cells. We can do 900,000 cells probably in a couple of minutes. So that's the difference here, is that we have the quantitation over looking at cells under a microscope. So what kind of samples can we do? Anything that you can get into a single cell suspension and is in between a certain size. So human cells from any part of your body, animals, plants, fish. I have to remember these pictures: algae, bacteria, yeast, and fungi, microparticles, nanoparticles, and beads. So all of this range I have whacked through a flow cytometer and seen the results, and some of them are quite interesting. And for purposes of safety, you and me go with the bacteria and fungi and algae and yeast, etc., because our samples can be biohazardous to other people. So that's where we go. So when you're looking at what it actually does, if you look at a cell there, on the surface of cell are structures that are called epitopes or antigens, and they recognize other cells and they interact with other cells using this. So we can put antibodies, and that's another word that you've probably heard on the news quite a lot to do with the coronavirus. So we can put on antibodies that recognize these epitopes and they bind to them, and attached to these antibodies are the fluorescent tags. And I'll put a picture on the right-hand side of some sea algae and a jellyfish. So these are some of the sources that we get the fluorescent tags from. They are also manufactured in labs and chemically, etc. But I think the natural ones are much more interesting and actually quite often a lot more stable and brighter than that. So we can look at the surface of cells, we can look at inside the cells, and we can look at inside the nucleus as well. So how a flow cytometer actually works, so you've got your cell there and it's got its fluorescent tag on it. You put it in a tube with a bunch of other cells, and then you put it onto a flow cytometer like this. Then you focus those cells, so they're going in single cells and they get hit by a laser like this. And then what we can measure is the light that scatters in front of that laser with a detector, and also the light that scatters at 90 degrees and the fluorescent tag through another detector. So this detector at the front is called forward scatter, and it's relative to the size of the cells, and the detector at 90 degrees, which measures the internal complexity, it's called side scatter, and also the fluorescence from that fluorescent tag that you've placed on there. Fluorescence, there you go. So what actually happens after that, this is a plot that you actually see out the end. So once those detectors measure that signal, they turn it into a pulse, and that pulse has an area, and then that area can be measured and placed as a dot on a plot. So that's what you've got here. So you can see on the x-axis of this plot is forward scatter, so that's relative to your size, and then on the y-axis is your side scatter, so relative to your complexity. And up on the left, you've got a picture of actual human white blood cells. So here you've got lymphocytes, and you can see they're literally just a nucleus with a bit of cytoplasm around it. So they are small and less complex, and they sit down here. And then you've got these bilobed monocytes, so they're much bigger, but not so complex as something like a neutrophil or an eosinophil, which is really massively complex. And then you've got these, all the sort of little fragments like red blood cells and debris and platelets and stuff, and they belong in the bottom corner. So looking at a real example, a good example and a relevant example, I think. You've got this group at the University of Modena in Italy, and they're looking at T cell activation in people, COVID patients with pneumonia. So you can see all the plots up on the right-hand side. You can see those dots. Every one of those dots is a cell, and as I say, they're measuring the fluorescence detection in the area and placing it as a dot on the plot. So they found that there was an activation of lots of different types of immune cells, and therefore they could then maybe get some targeted cytokine, which is your intracellular antibody therapy going. So it's a great paper if anybody wants to read that. So somebody asked what the coolest technology is. This is Fred and George. They are my cell sorters. They're named after the Weasley twins, of course. So if I just go back and you look at any one of those plots up on the right-hand side, you can see that there's some gates, which are if the bottom right-hand plot, it's got T, I think something about memory, SCM, sorry, gates down there. So if we wanted that population and we wanted to actually do something with it, we can then put it in one of these machines and we can actually sort out that population into a separate tube, and then you can take it away and do something else with it, look at its genetics or grow it further and see what it does when you do stuff to it. So that's what these two do. So they're great, but a little bit of a hassle, but I do like them very much. So there's so much that you can use flow for. There's loads in the clinical and loads in the research, and this isn't all. I'm not going to go through all of these, but it is really just a huge area. I think at the moment, I think there's about 30,000 papers a year that come out with some kind of flow cytometry in it. So it's a really relevant field for a lot of the areas. I found, especially when I worked in London, I found that I was getting people from physics department, from bioengineering, from chemistry, from normal biology. Anytime you do something to a cell, you want to see what comes out the other end of it. So we did have a lot of people from various areas. So I'm going to go into the questions now. So I think I have time. Please stop me if I'm wrong, if I have enough time to answer those and the ones in the chat. Somebody asked, in what ways is flow applicable to pathology? It's hugely applicable, especially to clinical pathology. So you have, especially in the hematology and immunology areas, with the diagnosis and prognosis of diseases like leukemia, flow is, basically a huge diagnostic tool for that. How has the work that I do evolved over time? It's evolved exponentially, and particularly in the technology. So when I started, the equipment that I was using was probably, like, the lasers I was using was probably about as tall as me, which is about 5'4" or so, huge water-cooled tanks, and we could look at 8 of those fluorescent tags stuck onto a cell. So now the lasers are probably about this big, and you can look at 40 plus colors. So the difference there is just enormous in terms of the evolution of the technology. My favorite part of my position is seeing people actually get results that they really want. And seeing people really happy about using flow. I say that because I had one lady come to me and say that she was terrified of flow cytometry, because there's giant machines. You walk in the door, there's these giant machines, they're looking at you and you're thinking, how can I break this, or something like that. So at the end, after a couple of months, she was like, I love this. I can get the results I need. And that's my favorite part of my position. Advice I would give to someone who wants to do the same thing or something similar, latch on to a really great mentor. I've had some really great mentors in my roles, and some people that have really inspired me to do a lot. So I've done that. And then finding yourself a great lab that supports technicians really well is really important. Would you recommend traveling abroad for your career? If you can, that would be really awesome. And my experience traveling abroad has been great. I've met many different people. I've met many different cultures. And it's really contributed to my career, as it were. So out of the countries I've had careers in, was there one that seemed to teach science better than the others? Oh, I could really put myself in it here, but I'm going to say no. It was very similar across the board. There were many similarities, I have to say. But the last bit here, throughout my career, what differences have you seen, how science is done through the world, and what stood out to you the most? Definitely funding. So in my home country in New Zealand, there's very little funding for science reasons and science purposes, I should say. And there are many great scientists there that have had to leave and go to other countries, including here in the States. In England, there was quite a lot of funding, but it's kind of directed down different pathways, like HR pathways, and maybe not to the facilities that need them. The small experience I've had here, so I've actually only been here since February last year, and the funding has been fantastic. I must admit, I really appreciate the donations from people. That means that we're able to buy equipment and everything. So probably the funding. So my favorite question: do teaching methods I am developing, including teaching her students how to communicate complex ideas in simple ways. And then that this person found that most scientists and people in this field are very knowledgeable, but never know how to communicate their ideas to people. That's exactly it. That's exactly why I kind of looked at doing a diploma myself, so that I could teach differently. And one thing about this was there was I had a lecturer in evolutionary biology, and he was American, as it happens. But this is at university, he was obviously very passionate and very knowledgeable about his subject. But I think in the end, about 70% of us failed, because we couldn't understand him. We couldn't understand what he was talking about, and his method of communication was really poor. So that's what I really keep in mind. I make sure that people are understanding what I'm teaching. So I give them a lot of quizzes. I question them. I don't know if they like me too much for that, but that's fine. I make sure that I have teaching resources that encompass everybody's learning differences or preferences or styles. I know that I learn very visually. I learn by looking at pictures and diagrams and things like that. I find it very difficult to sit and read a scientific paper. So I make sure I have things for people like myself and for people that learn well through reading scientific papers. So really that's really important. And yeah, just offering a lot of feedback opportunity, because I think the way that teachers develop really well is by hearing feedback from their students. So I do make sure that I do that as well. So I'm going to look at the questions in the chat. I can. It has actually affected it quite a bit. We got shut down in March last year. I started in February and we got shut down in March, and no work was happening up until June. And then we've slowly started to go back. I think we're just slower, to be honest. And a lot of people at the Gladstone Institutes have changed to working with the coronavirus. So we have to be really careful and get tested all the time, but that's really about it. So we're a little bit slow at the moment, but I'm guessing that won't be the case soon. Did I always wanted to do lab work? I had no idea. I really was just at the beginning, I was just working in it just so that I could get into work, because I was in the situation where I had to pay my own bills and kind of support myself. So I just needed to work, really. I think as I got a bit older, I could see that the technical pathway was for me. Have I considered a different path? I thought I was thinking about being a teacher. I always wanted to be an astronomer, but I was rubbish at math, so that was out. That's about all, really. Explain how I got into flow even after majoring in microbiology. So my first job was that technician looking at the arthritis patients, and I actually got that job through someone I know, because sometimes that's who you know, not what you know, unfortunately. And so I got that through them. And then I went into another job, and while I was in this job, I really didn't like my boss. I thought they were not being very good for the lab, or very good for our career development. So I saw this job advertised that had a lot of what I did in it, but this thing called flow cytometry as well. And I had no idea what it was, but I thought, oh, that's fine. I'll just apply for it and see what I got out of it, get out of it. And I got hired for it and I got trained. So that's how I got into flow. So is there any more questions there?
There's one hand up. Russell has a question.
Russell, so yeah, go ahead. Yeah, I actually enjoy it. I mean, there's some people that really take to it really very quickly, and there's other people that may need a bit more help. And it's been really great for me to be able to make sure I really help those people that maybe don't flick onto it very quickly. I have to remember, one thing I have to remember, and I think that's applicable to anybody that gets to about my age and has been in the industry for a long time, you have to remember what it's like to not know anything about the subject that you're teaching. And so putting yourself back in that position can be really hard, but it's also really helpful. And just chatting to the person that you're teaching and sort of making sure that they understand is really important. Currently do a lot of research with HIV, because we're a big HIV research institute, and just I think there's some research came out recently with flow, just looking, because in these patients nowadays, HIV is not a death sentence, if I can say that. You know, you can live long term with HIV, but you generally live with latent pockets of it. So we're looking at the cells that have the HIV, that are these latent pockets in there. A lot of coronavirus research, obviously, and the Gladstone also does a lot of neurological and cardiology research, which we're involved in too. Right, I think that's all. Is there anybody else? I don't know if anybody else has their hands up. Hang
I
on.
think there's maybe two things we can cover really quickly.